Excellent. So I've had a lot of questions about this figure that you see on the screen. So I know everybody's confused by it, and with good reason. I was confusing about it. So I had a group of you after class that came up to me and said, oh, that thing's labeled wrong. The parallel is anti-parallel, and the anti-parallel is parallel. And I said, oh, no, it's just part of a protein. In fact, it is part of a protein. You're not seeing two intact, separate strands. The fact that you have the nitrogen at the bottom on one side, let's say over here. Ooh, look at that. And I have another one. I have the oxygen on the other side over here. Okay. That just shows up on one, doesn't it? That's interesting. Okay. Does not mean it's anti-parallel. Okay. So you're just seeing a part of a protein. It's how we orient it. But then I went and I looked at it, and then I realized that it is mislabeled. Okay. So, and the reason is if you look at the orientation of the NO, let's say this, compared to the equivalent here, they're in opposite orientations. So, what's labeled on there as parallel is actually anti-parallel. What's labeled as anti-parallel is parallel. I should listen to my students more often. I know that. Okay. So, I apologize for the error, and you are correct. So, the parallel is anti-parallel. The anti-parallel is parallel. Okay. The other thing I had somebody before class, just 1 second, just before class comment on that I wasn't aware of, and that is if you look on the syllabus for page numbers, they really won't make a lot of sense. Okay? So the syllabus page numbers, I need to change. They were from an old book. So the correct page numbers are on the schedule page, as I hope that you all are using. And you had a question over here? How I knew it was parallel, anti-parallel? So in any of these, you would have to look at the relative orientation of the atoms. So you see in the middle, for example, here, you see that you have orange and, oh, that's white. I don't want white. You see that you have orange, purple, black, right? And you see orange, purple, black. That would be parallel. Okay? And this is parallel, like I said, because you have carboxyl ends lined up the same end as the amino ends. All right. So, all right. Yeah. I'm sorry?
The page numbers of the other textbooks, like the physical editions, can you leave that on the website as well when you update it?
Could I leave the Stryer page numbers there? To be honest, I don't want to encourage people going out and buying Stryer. So if you want it, send me an email and I'll send them to you. How about that? Okay. Okay, so let's pop out of that for a second. And I promised you a song at the beginning of class, and we will have a song at the beginning of class. And we're going to have one at the end of class if we get through things okay. Ooh, let's clear that. All right. This guy is a real popular song. It's a real easy one to sing, and I want to hear you sing loud and clear. It's called "Oh Little Protein Molecule." And it should be starting. And it's not starting. And why didn't it start? Let's exit this. Maybe the audio. Who knows? Oh. We'll try it again. We'll try it again. Oh, little protein molecule. Oh, little protein molecule, you're lovely and serene, with 20 Zwitter ions like cysteine and alanine. Your secondary structure has pitches and repeats, arranged in alpha helices and beta pleated sheets. The Ramachandran plots are predictions made to try to tell the structures you can have for angles phi and psi. And tertiary structure gives polypeptide zinc Because of magic that occurs in protein folding, a folded enzyme's active and starts to catalyze. When activators bind into its allosteric sites, you'll learn this later. Other mechanism control the enzyme's rates by regulating synthesis and placement of phosphates, and all the regulation that's found inside of cells reminds the students learning it of pathways straight from hell. So here's how to remember the phosphate strategies. They turn the GPBs to A's and GSAs to B's. And we'll talk about all that later. So, okay. That was a long one, that's right. Okay. By the way, if I have singers in here who are interested in singing, the College of Science is putting on a thing this year with the College of Liberal Arts called SPARK. Anybody heard of SPARK? Demonstrating scientists and liberal arts people working together and doing some cool arts projects. I was asked to assemble a choir to sing metabolic melodies. And I'm looking for people who would like to participate in that. And if you're interested in that, come see me after class. I'd love to have you. I really need singers. You know I can't sing. And I won't sing, well, I'll have the choir sing. So if you're interested in that, please come see me. Okay. Shh. The turns I talked about last time, I'm not going to go through that again. But turns, we remember, are fairly short structures that involve a change of direction of a helix, or of a protein sequence. This is a nice protein, looking at it sort of end on, that illustrates some of the structures that we've seen. And now we see that this is a protein that's not a fibrous protein. Fibrous protein, we had a helix that just went on and on and on and on. And I told you that a fibrous protein had primarily primary structure and secondary structure. It had very little tertiary structure. Well, tertiary structure actually arises from turns. Turns cause a change of direction. They cause a bend. They cause proteins to have the structures that they do. We can see in this protein that it has some alpha helices. The alpha helices, of course, are shown in purple. We see it has some beta strands located within it, shown here in yellow. And we see that those strands are oriented in a sort of a sheet-like fashion. We also see turns. You see some turns down there at the lower right, and those turns are relatively short regions that are causing a change in direction. There's another structure that's on here that's labeled random coil that looks kind of like a turn, and if you want to think about it that way for our purposes, I won't have any problem with that. But technically, a random coil is not a turn, but it's a longer stretch that will have more of a randomness in terms of its orientation. But this is what we call a globular protein. A globular protein has three-dimensional structure, and it has three-dimensional structure because it has, yes, thank you. It has three-dimensional structure because it has those turns within it, and also the random coil, as you can see here. Well, globular proteins get the way they are by folding. I showed you earlier an illustration about how folding occurred, and I pointed out to you that folding in an aqueous solution occurs so that the hydrophilic amino acids will be on the outside facing water, and the hydrophobic amino acids are on the inside avoiding water. We'll see some things that violate that rule today. But suffice it to say that an unfolded protein gets folded, as we can see here. One of the things that happens frequently with folding is folding actually happens as the protein is being made. So we see folding start almost immediately. So if I take a completely folded protein and I unfold it, the folding environment that it finds, if it tried to let it refold, most proteins won't refold properly if we do that. There are many reasons for why that's the case, but one of those is the fact that when it's folding in a completely intact form, that's different from the way it started folding, which was folding as it was being made. Okay? So there's too many confusing directions for it to go if everything is there to start with. So keep that in mind when I talk later about the fact that proteins unfolded usually can't be refolded properly. Unfolding of a protein can happen as a result of a variety of things, but one of the easiest ways to unfold most proteins is simply to heat it. If I take and I heat a protein, I break hydrogen bonds, and those hydrogen bonds are important for the overall three-dimensional structure of a protein, helping to stabilize the secondary structure and also helping to stabilize the tertiary structure. We'll see that there are other forces that help to stabilize tertiary structure as well, but this schematic shows that heat can have a pretty good effect, or a pretty big effect, on the structure of a protein. It's because of this ability to denature proteins with heat, as I mentioned earlier, for the reason we cook our food. We destroy the proteins in the bacteria that have those proteins that are destabilized by heat, and therefore we kill the bacteria. Now, tertiary structure is more complex than secondary structure in terms of the things that hold it together. And that's because tertiary structure is not a regular structure. Tertiary structure is not restricted by a helical nature, which only allows for close interactions of things close to each other. We saw in that protein that I showed you that different regions of a protein could interact with each other depending upon how the protein actually folded. So we're going to see a variety of forces that come together to hold tertiary structure into the proper configuration. And I want to emphasize that these forces are very important for the protein being functional, because structure is necessary for function. You destroy structure, you destroy function. So if you overcome these forces, you will overcome the forces stabilizing structure and function. Okay. Well, hydrogen bonds you've seen before, they arise from unequal sharing of electrons of atoms involving hydrogen. And they happen primarily with hydroxides and sulfhydryl groups and amines, as we will see in proteins and various other things. Hydrogen bonds, of course, can happen in a variety of ways. Here's a good example of two carboxylic acids. This is acetic acid that are interacting through hydrogen bonding, as you can see here. Now, another force that helps to stabilize protein structure, that is tertiary protein structure, are disulfide bonds. I need to show you those. So, imagine I have a protein that has two cysteine amino acids within it, and they've been brought together into close proximity. Now, I need to also remind you of something I said briefly earlier, and I probably didn't make as clear as I should have, and I'll make it clearer here. That is what the term tertiary structure actually means. Okay? I said that primary structure was the sequence of amino acids in a protein. I said that secondary structure was a regular structure that appeared as a result of interactions between amino acids close, that is, about less than 10 amino acids away from each other. Tertiary structure, by extension, arises from interactions between amino acids that are not close in primary structure. That is, they're more than 10 amino acids apart from each other. Right? So we can imagine that a protein that had two different cysteines, that were one was located on this part of the protein over here, and another one was located way over here on another part of the protein. When the protein folded, these guys were brought into close proximity. Everybody with me? So you can envision that these parts that started out very distant from each other ended up being very close to each other, and when they ended up close to each other, those two can form a covalent bond between them called a disulfide bond, which is what you see on the screen. And that's a sulfur-sulfur bond. It is a covalent bond. Remember I said that covalent bonds are much stronger than hydrogen bonds, and so disulfide bonds are very important in helping to stabilize tertiary structure. Yes, Maddie. What's the difference between supersecondary structure and tertiary structure? I had a bunch of people in my office the other day asking me that. Don't worry about it. Okay? No, I'm serious. I don't mean to blow you off. But I use the term supersecondary structure more to arrange secondary structures together like in a sheet. But they also arise from things that are farther apart from each other. So don't get hung up on the terminology. I'm not going to ask you to distinguish for me supersecondary versus tertiary. Okay? But I do use the term to talk more about how the individual secondary structures arrange. Okay? Yeah, I didn't mean to throw it out there to confuse people, because I know it confused several people who came and asked me about that. All right, what I want you to take here is this disulfide bond. Okay? The disulfide bond is a result of an oxidation. You know it's an oxidation by virtue of the fact that you see 2 electrons flying away there. Okay? Sulfhydryls turn out to be very reactive. You put them in close proximity, they like to do exactly what you see here on the screen. The result of that is a set of 2 amino acids joined together. Some people call that structure cysteine, C-Y-S-T-I-N-E, instead of cysteine, which was C-Y-S-T-E-I-N-E. So it has a sulfhydryl, it's cysteine, it's a disulfide bond, it's 2 of them joined together to make cysteine. Well, this figure I happen to like a lot because it shows all of the different kinds of forces that can help stabilize the tertiary structure of a protein. We see that a tertiary protein, we see that a globular protein that has tertiary structure, as you see on the screen, has secondary structures within it. You see that on the right. There's an alpha helix, and there's also a beta strand that's there. And you remember that hydrogen bonds hold those structures together. And hydrogen bonds can also hold together other parts of a tertiary structure without being in an alpha helix or without being in a beta strand. You see the disulfide bonds that I just showed you on the last slide. And the disulfide bonds, of course, are the covalent forces that help hold things together. You know also that plus charges attract minus charges. You know that plus charges repel plus charges and minus charges repel minus charges. And so ionic forces or ionic bonds also influence, stabilize tertiary structure. So it's this point where we think about, well, if I change the pH and I change the charge of the side chains of amino acids, I'm going to change some of the forces that are stabilizing this protein. I might think then that if I change the pH drastically, I would denature or unfold the protein. And with most proteins, that's exactly the case. That's exactly what will happen. So it's happening because we're changing the charge on those things that are ionic in nature. If you look up above on the top there where you see the thing that says hydrophobic bonds, okay, that's a really important one, very important structure in the stabilization and also the formation of tertiary structure. Remember that hydrophobic amino acids, or amino acids that have hydrophobic side chains, don't like to interact with water. If I put oil in water, what happens? Oil will associate with itself and separate away from water. If I take amino acids that don't like water, and they can find each other, what are they going to do? They're going to associate and avoid water. That is a force that helps to drive the process of folding. The desire of those side chains of hydrophobic amino acids to avoid water causes them to associate with each other, and that's one of the forces that starts the process of folding. Okay? It's also a force that helps to stabilize folding, because once they've gotten together, they like to stay together. Not a problem, right? So I've talked, I'll say a word here about these, because another thing that we do with respect to bacteria and killing bacteria is washing our hands. Washing our hands, we use soap, and when we use soap, what are we adding to our hands? We are adding amphiphilic molecules, right? We talked about those fatty acids that have a nonpolar part and a polar part. The nonpolar parts, guess what they do? They stick themselves into hydrophobic things. And some of those hydrophobic things are the side chains of amino acids that you see here. You can imagine that when you wash your hands with soap, the soap will disrupt the forces, the hydrophobic forces that help to stabilize proteins. Okay? And in fact, that's what happens. You wash your hands with soap, you are denaturing unfolding proteins because you're disrupting those hydrophobic forces that help to stabilize them. So again, we see a practical thing from a health perspective relating to why we do practices that we do, and they come down to what we're doing to the proteins in bacteria that would otherwise do us harm. The last force seen on the screen that I'll talk about with respect to stabilizing tertiary structure is that of metallic interactions. Metallic interactions can help stabilize the overall structure of proteins. And while they're important, we won't talk about them a lot in this class. We'll talk about them just a little bit. Okay, questions about that? Clear as mud? Nobody's awake today. It is Friday. You can always tell. Oh, yeah. Are the hydrogen bonds between amine groups? Is that what you're saying? So I think you're asking me not about the alpha helix, but rather about the beta strand. Is that what you're asking about? Okay. So the beta strand has those same forces that I showed you earlier when we had the parallel and anti-parallel. If I were to show you this, would you tell me this is parallel or anti-parallel? What's that? It's actually anti-parallel. Okay? Imagine that you started, okay, at the lower right of the coil of the alpha helix, and let's say that is the amine end. That means that moving upwards, we have amine to carboxyl direction. Bending around, we have amine to carboxyl direction going down. And when we bend back up, we have amine to carboxyl direction going back up. They're opposite. That's an anti-parallel orientation. Everybody understand that? Okay. So whenever you see something bend back on itself like that, it'll always be oppositely oriented. Yes, question? Okay, so I think I understand the question in SMAC, right? Yeah, I think the question I understand is, do these hydrophobic bonds form later? Is that the thing? And that the other secondary structure things are forming first? Is that what you're saying? It's a good question. This figure might imply that, and I don't want you to read that from this figure. However, since secondary structures happen very close to each other, they will, in fact, happen first, because they happen before the overall thing gets a chance to fold back on itself. This figure, I don't mean to imply that with this figure, but the observation that you made is correct. Okay. Okay. Where am I? Oh, there we go. Label, 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 label. Okay, good. All right. If we want to break or denature or unfold a protein, I've already led you to some of this already. We need to break the forces that stabilize them. One of them I haven't talked about is how do we break disulfide bonds? Right? How do we break disulfide bonds? Well, the cell has a way of doing that we'll talk about later. So what I'm going to talk about here is not what a cell does. It's what a scientist does in the laboratory. Scientist breaks disulfide bond with a chemical called mercaptoethanol or a different chemical called dithiothreatol. Both of them will break a disulfide bond. Now, when I say break, all it's doing is it's converting the disulfide back to 2 sulfhydryls. But it no longer has a covalent bond between them. That's an important consideration. Because if that covalent bond between them, the disulfide bond, is an important structural component of tertiary structure of a protein, I have just destabilized that protein. Now, just because I destabilize a protein doesn't mean it unfolds. I may have to do other things to it. All right? But suffice it to say, if I destabilize this building, I don't want to hang around inside of it to wait to see if it falls when I destabilize it or not, right? Yes, ma'am? Very good question. So her question is, can you partially destabilize a protein by only destabilizing certain parts of it? And the answer is you can. Okay? However, how any given protein responds to that depends completely and uniquely on that protein's overall other structure. So you can imagine some proteins might have a lot more hydrogen bonds stabilizing them than a different protein. So if I destabilize the hydrogen bonds in a protein that I could do relatively easily, that might be a bigger factor for one protein than another. Okay? Good question. Okay, I mentioned detergents. Detergents disrupt the hydrophobic bonds within a protein. Okay? Heat breaks hydrogen bonds. Another thing that breaks hydrogen bonds, I didn't put it on here, is urea. Urea in high concentrations will disrupt hydrogen bonds. Okay? We use it in the laboratory a fair amount sometimes to disrupt hydrogen bonds. I mentioned pH earlier because pH, of course, changes charge of side chains of amino acids, and since some of those are involved in helping to stabilize or hold a protein in its tertiary configuration, pH can be a big factor in that. Chelators. What's a chelator? So a chelator is something that binds to a metal ion. There are a variety of chelators that are out there. One that we commonly use in the laboratory is called EDTA, ethylene diamine tetraacetic acid, but you don't need to know that. Okay? EDTA will bind to magnesium ions, for example. If magnesium is important in the overall structure of a protein, that can help, again, to destabilize the overall protein. Okay. Well, one of the proteins that we talk about, this is a really good example. It's a good example and it's a bad example. It's a good example because it illustrates the importance of disrupting all the forces in this particular protein to be able to denature it. It's a bad protein in that it's unusual. It gives us the impression that we can refold all proteins back into normal functioning form. Most proteins cannot be refolded properly back into their functional form. But this one can. And this one has some interesting properties, so I want to go through and tell you about it. This is a protein called ribonuclease, or as we call it more commonly, Rnase, A-S-E. Rnase is really interesting because Rnase is very, very stable against most of the forces that I've already described to you. If I take Rnase, for example, and I put it into a tube, and I boil it, I discover that there's still some activity there. Whoa. Okay? It tells us, first of all, that the hydrogen bonds aren't the most important force that's holding Rnase together, and I can treat it with a variety of agents, and I still have some activity associated with it. And only when I take Rnase and I treat it with beta-mercaptoethanol and heat, or with urea, do I discover that I lose all of its activity. Now, that information I just gave you there should tell you that there's disulfide bonds that are playing very critical roles in the tertiary stability, tertiary structure, tertiary stability of Rnase. So to denature this protein completely, I not only have to break the hydrogen bonds, which in this case I'm breaking with urea, but I also have to break it. Also have to use mercaptoethanol to break the disulfide bonds. Everybody with me there? So I could use heat, I could use urea in the same way. They both break hydrogen bonds. If I just use urea or I just use heat, I don't see that Rnase loses its function. Oops. Yep, I didn't want to do that. All right. Now, Rnase, and there's a problem set that I gave you. Rnase is an interesting protein in the sense that if I take unfolded Rnase and I put it back to room temperature and I get rid of the urea, for example, what I discover is that there's a decent percentage of Rnase that will regain activity. It lost, all of its activity was gone, and some of it will become active after I remove that urea. And when I remove the urea, and my mouth is not working today, some of it will gain activity when I remove the urea and the mercaptoethanol. Now, there's a curious thing, and it's in one of the problems that I gave to you, that if you allow it to renature, which is what's happening here, it's regaining its activity, in the presence of a little bit of mercaptoethanol, you get even more activity. You get even more activity. Now, how in the world is it that this thing that's breaking disulfide bonds is allowing for more activity. Because Ahern, you just told us that to destroy the activity, we had to add mercaptoethanol, and now you're telling me to regain the activity, we have to add mercaptoethanol. Well, before we think too much about that, I want to point out something to you that is a very important thing that you probably didn't think about as I was telling you this. The very important thing is the very fact that I can unfold Rnase and it refolds on its own to any extent. That means that all the information for folding is contained in the primary sequence, because that's the only thing that's there. There's nothing else folding it. It's folding itself. So primary sequence, I told you earlier, determines all the properties of a protein. It also dictates the folding process. And that's why if I change the amino acid sequence, I will get a protein that's differently folded. So Rnase is the best proof of that right there. Now we'll take a minute here and think about that observation I just gave you. Some of you I sort of gave the answer to in my office yesterday, so I'll ask you not to divulge that here. But I'd like to think for a minute how it is that a little bit of mercaptoethanol helps this process to work. Any thoughts that people have in the class? Yes? Is it what? Is it a catalytic process? The answer is no. Nice try. Yes? That's a very good observation. So she said, is it possible, I'm going to reword what you said, but basically is it possible that the hydrogen bonds play a role in that overall folding process, and it's helping to get it into the proper configuration before the mercaptoethanol, before the disulfide bonds form? There's a lot of truth in that, but that's not the answer to the question that I asked, but you're right. You're exactly right. Why does mercaptoethanol make a difference? Yeah. Okay, so what's your name? Gwen. Gwen's on the right track. She says a little bit of mercaptoethanol only allows some of the ones to be broken and maybe not others. But that's partly right, but it's not quite the right answer. One other thought. Anybody? Yeah. Ah! Excellent. Excellent. What's your name? Evan. Evan just basically hit it on the nose. There's a lot of sulfhydryls there. Those sulfhydryls are flailing around in the process, and you might imagine that there will be some misdirection. There's going to be some incorrectly bonded sulfhydryls that will happen. He called it misfolding, and that would certainly lead to misfolding, right? And if that happens, then once you get two things that are incorrectly bound, you'll never get the proper protein. But if you have a little bit of mercaptoethanol, you can release that and give it a second chance to fold properly. You put too much in, you won't get it, because nothing will form. But a little bit allows those accidental ones to unfold, or to unlink, and then the proper folding to occur. Very good, very good. Okay. Well, there's the folding process that I talked about before. When we think about folding, folding's a pretty amazing thing, okay? Folding's incredibly complex. When we first started studying folding back in the 60s, people said, oh, well, we'll figure it out with computers. And folding is an energy and entropy driven process. Okay? For folding to occur, it needs to be energetically favorable overall, and the ideal place for that folded state is at the very bottom of this energy diagram. But you see that if we start at the unfolded state at the top and we get to the bottom, it's not a straight fall. If we go to the Grand Canyon and we jump off of the cliff, we don't necessarily end up in the Colorado River. We end up on some cliff that's far a ways down. That's what can happen to a folded protein. It can get stuck in any of these individual gaps that you see in there. And if that happens, it doesn't get a chance to fold properly. The properly folded protein will bounce, 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 all the way down until it folds properly. But proper folding doesn't always occur. We know that, and we know that the consequences of proper folding not occurring are very large. Okay? We'll talk about those in just a second. But before I talk about those, I want to talk about structures in tertiary structure of proteins. We see many proteins have similar structures, and they have similar functions, obviously. We can imagine that cells have a common structure for binding ATP. ATP is used universally by cells. Many proteins need ATP. We might expect that all proteins that bind ATP have a similar ATP binding structure. That's one example. And so what I'm defining for you are what we call domains. Okay? Structural domains are common features we see across many proteins that have similar function. One of these is called a leucine zipper. We'll see an example of it right here. Leucine zipper happens as a result of two helices that have leucines sticking out, and they interact in a zipper-like fashion that you see here. You see a bottom strand shown in yellow and a top strand shown in purple with their leucine side chains interacting. And it literally forms a zipper. That zipper functions to hold those things together. We see leucine zippers in many DNA-binding proteins and in some protein-protein interactions. There's a leucine zipper that's binding to DNA, and you can see the zipper function really working very well to hold that thing together. Helix-Turn-Helix is another protein structure that we commonly see that's found in DNA. And by the way, you're not going to recognize, I'm not going to ask you to draw these or something like that. I might expect you to know what a leucine zipper is to be able to tell me, for example. A helix-turn-helix is a common structure in protein that binds the DNA, and we can see the individual parts of the protein are interacting with what we call the major groove of the DNA, as shown here. Zinc fingers are parts of a protein, and these are all parts of a protein. These are not proteins, but parts of many proteins. Zinc fingers are parts of a protein that have a zinc, and it causes the protein to sort of bend around that zinc, and it forms, literally, a finger. You can see it poking itself into various things, again, commonly DNAs. Another common structure, another common domain we see across many proteins is known as an Sh2 domain. This is mostly a protein-protein kind of interaction. But again, a common structure we see in many, many proteins. These proteins are commonly involved in the process of signaling. And finally, another signaling domain we see is called a plextrin homology domain, and you can see that these are in proteins that are interacting around a membrane. You can see the membrane is the blue line in the plextrin domain region in the bottom on the right. Well, as I said earlier, folding errors cause problems. You know of some of those problems. Everybody's heard of mad cow disease, right? Not something to mess with. Mad cow disease is not caused by a virus. It's not caused by a bacterium. It's actually caused by an individual's own proteins. Caused by a misfolding that occurs in brain protein. That brain protein leads to the formation of what are called plaques. And those plaques destroy nerve cells. A person who has this, or more commonly a cow who has this, loses all mental function because you destroy the nerves in your brain and you don't have anything to function on. This folding error starts with a normal protein, PRPC. Has a structure shown here in a circle. Misfolding of one of those. This is a little scary how tenuous your life is, folks. Misfolding of one of those causes that one misfolded one to cause the others to misfold. And that's what you see happening here. PRPSC, that's the misfolded protein. You can see that it's inducing normally folded protein to misfold into the triangular shape. And that triangular shape makes long polymers that creates the plaques that I described to you. Okay? Mad cow, pretty devastating disease. Mad cow disease is transmitted from cow to cow. It's transmitted. We don't completely understand how prions These are called prions. I should say that also. These are called prions. These misfolded proteins are called prions. P-R-I-O-N. Okay? Humans have an equivalent disease called Creutzfeldt-Jakob. I'll put that in the highlights so I won't spell it for you here. Creutzfeldt-Jakob disease. That also involves the same protein. It involves misfolding. Okay? There's speculation that can a person get Creutzfeldt-Jakob by eating mad cow protein? We don't know the answer to that. There was a period in England in the late 1980s where there was a large incidence of mad cow disease, and the beef supply was not screened very well. Question? Oh, yeah. So Kuru is a related disease. They're all similar diseases. Yeah, yeah. So they all arise from prions, and they all arise from misfolding of the same thing. Yep. Okay. So that back in the 80s, they had. Did you have a question? How does it induce the misfolding of others? There you go. That's the best answer I have for the question. It's actually, as you can imagine, a complicated question. Okay? But suffice it to say that you could imagine that you've got a sort of a template that one of them forms and the other one binds to that template, and as a result, it gets changed accordingly. But it's a much bigger question than I can answer in a simple fact. In fact, it's not even completely understood. Yeah, question? Question over there. Yes, I think the originating question is over here. I'm sorry? CTE being? I'm not a physician. I don't know the answer to that. With concussion, you usually have very different kinds of damage that's there. So my gut feeling would be no, but I'm just not a physician. I can't answer that. I don't know. Okay. Anyway, I want to tell you the story about the. Any other questions? That's good. I like the questions. Don't hesitate to ask questions. The beef supply got, they think, contaminated with some of the mad cow meat. Incidence of Creutzfeldt-Jakob in England went up about 3 years later with an odd form of the human variant of Creutzfeldt-Jakob. The thinking was that, in fact, it was something that people had eaten. And you might say, well, how can you eat that? You just cook your meat and you're fine, right? I've got really bad news for you here. To unfold a misfolded prion, takes a temperature of about 700 degrees. Nobody wants their meat that well done. That means normal cooking will have no effect on such a protein. Now the question is, can you eat it and then have it get to your brain, etc.? That's the $64,000 question people argue over. I'm not going to wade into that debate. All right? This is where the vegetarians go, right? Yeah. Okay, so prions are self-replicating. They're self-replicating in the sense that they just induce the misfolding of properly folded other proteins. These create structures we call amyloids, collection of improperly folded protein aggregates. And amyloids, we know, are factors in several diseases. When they're misfolded, they're insoluble, and there's 20 human diseases. They all involve neurological problems. They include Alzheimer's disease. They include Parkinson's disease, Huntington's disease. And I've labeled in each case the problem protein involved in the amyloid structure. Rheumatoid arthritis, fatal familial insomnia. And to be honest, I don't know that one very well. Okay? I have insomnia, but I don't think it's that. I shouldn't joke. Okay. Protein processing. Folding's important. Cells, therefore, have something to ensure they don't have problems with folding. They have a structure. They have a couple proteins called chaperonins. Chaperonins look like this. And they're little chambers that are found in cells that allow proteins to properly fold. not all proteins are folded in chaperonins. But some are, and some need that environment in which to fold. Now, I'm going to give you an example, and then I think I'm going to cut to the song for the day. Oh. All right. There we go. There we go. There's my thing. All right. Imagine that you're making a protein, and it's got a long stretch of hydrophobic amino acids. What do you suppose those hydrophobic amino acids are going to do? Well, they want to interact with each other, right? Except for the proteins being made, and they don't really have an exterior and an interior of a protein at this point. If it's a fairly long stretch, you start thinking, hey, we got a problem, right? Well, proteins aren't made in isolation. One protein being made here might be made adjacent to the identical protein being made over here. So we have two copies of the same protein. In fact, they're usually made in the same place. This guy over here that's being made has this little region that's very hydrophobic that's coming out. And guess what? This guy over here has a little hydrophobic region coming out as well. Won't they like each other? Yeah, they will. Well, putting them together isn't necessarily the way those proteins are designed to function. So now you just made a misfunctional protein. You've made a misfolded protein that, for the most part, the cell's probably just going to throw away, and that's that. But cells don't like to throw things away. So rather than encouraging misfolding or encouraging things that have large hydrophobic regions from having problems, cells have chaperonins. So instead of having a chance to interact with each other, a protein that needs help folding gets synthesized directly into one of these things. And then it doesn't have other environments that it can interact with. It can interact with itself and properly fold. Chaperonins help proteins to fold properly. There are some chaperonins that are actually involved in helping proteins to fold after they have misfolded. Heat shock. Anybody hear about heat shock proteins before? Heat shock happens when a cell has its temperature elevated very slightly, not enough to kill it, but in some cases enough to denature part of a protein. In that case, cells will make heat shock proteins to help the cell to recover, and those heat shock proteins include those proteins that make up a chaperonin. The GRO-EL, the GRO-ES, are the names of the proteins that make up a chaperonin in E. coli. So an E. coli chaperonin contains GRO-EL and GRO-ES. All right. The one last thing I do need to say is about proteasomes, because it's relevant for our song. Proteasomes are complexes that break down proteins. Cells don't have proteins floating around forever. It's important to be able to break them down, especially if something has happened to them. So proteasomes play that role. They break proteins down in great big chunks, down into little chunks of about 8 amino acids or so. I'll talk a little bit more about the proteasomes next time. You can see proteasomes right there. They're found in every cell on Earth because cells have to recycle those proteins. And that's what they're doing. They're breaking it down so that they can use the amino acids within them. All right, I do have a song for you. I'm going to stop at this point, and we'll do that song. We'll pick this up at the same place next time. The song is called My Old Enzymes. It's to a tune you'll recognize. Please join in. Whene'er my proteins go kaput, if they are past their prime, the cells will act to soon replace all of my old enzymes. They know which ones to break apart. Ubiquitin's the sign, a marker for proteasomes to find the old enzymes. These soon get up and then cut up in pieces less than 9. More chopping yields the single ones, building blocks from old enzymes. So in a way the cell knows well of father time, it's true. amino acids, when reused, turn the old enzymes to new. Happy Friday. Happy